Welcome to the lecture series on food microbiology. So far in this series, we have seen, depending upon the criteria of food we are talking about, what are the spoilage organisms and how we can extend the shelf life and improve in such a way so as to reduce the spoilage. In the coming series of lectures, we are going to see how to assess the safety of food and what are the factors assessed for quality control. Now, when you are talking from a food industry's point of view, there are two major aspects. One, the quality of the food, irrespective of the batch or the day you are going to market it, it has to have the same flavor, shape, whatever we talk about, it has to be the same. We call it as the quality control. So that has to be the same. And two, the safety of the food. It should be free from microbes, both pathogens as well as spoilage organisms. So how do we assess this? This is what we are going to see in the next couple of lectures. In terms of the microbiology of foods, the term quality, it comprises of three aspects. One is safety. A food must not contain levels of a pathogen or its toxin likely to cause illness when the food is consumed. Meaning, there can be microbes, but the level of the microbes should not be very high so as to cause it to be a harm to the consumer. And also the toxins, in many cases it's not the bacteria but the toxins which cause food poisoning, that toxin should also be present. You have what you call as a threshold level. So as we already discussed, whether to accept or not accept, that would be the deciding factor. Acceptability and shelf life. A food must not contain levels of a microorganism sufficient to render it organoleptically spoiled in an unacceptably short time. So when the microbe level in the beginning itself is high in the food, the chances of it getting spoiled very fast. Even if you say that you can keep it for 2 to 3 days, when the microbial level is high, the food will tend to get spoiled within 1 day, maybe less than that. Then the organoleptic characteristic, that is the nature, the flavor, the aroma of the food gets spoiled. So it should be acceptable by the consumer and it should not compromise on the shelf life. Those are two major aspects which we talk about there. Consistency. A food must be of consistent quality both with respect to safety and shelf life. So batch wise, day of production wise, year of production wise, whatever be the aspect you are talking about, from a particular company, when we are looking for a particular brand, that should be the same, that should remain uniform throughout. That is what we call as consistency. The consumer will not accept products which display large batch to batch variations in the shelf life and cannot fall ill every time a particular food is consumed. Regulatory bodies and the food industry are the two main groups most actively interested in determining and controlling the microbiological quality of food. The regulatory authorities must do so to fulfill their statutory responsibility, and this responsibility is to protect the public from hazardous and or inferior goods. So one aspect is the regulatory body, the other aspect is the food industry. Both work together to maintain a set standard so that the public, those who consume this, will not either be have inferior goods or they will not fall ill because of consuming a food which is not safe to consume. To distinguish food of acceptable quality from food of unacceptable quality, it requires the application of what we are calling as microbiological criteria. So this becomes very important, whether the food should be accepted by the consumer or not accepted. It needn't be even the consumer, it can be from the wholesaler to the retailer. So the first level acceptability or non-acceptability comes there. So for that we fix 3 microbiological criteria. There are 3 different types of microbiological criterion which has been defined by the International Commission on Microbiological Specifications of Food, otherwise ICMSF. A microbiological standard is a criterion specified in a law or a regulation. So the first is standard. So this we regulate by a law. We are saying that for this particular food, this much amount of microbe is acceptable. And what is the type of microbe? That depends. That acceptable level depends upon the type of the microbe we are talking about. This is a legal requirement that foods must meet, and it is enforceable by the appropriate regulatory agency. So irrespective of the country we are talking about, this standard is regulated by a law and has to be accepted by all food industries. A microbiological specification is a criterion applied in commerce. This is a contractual condition of acceptance that is applied by the purchaser attempting to define the microbiological quality of the product or the ingredient. Failure of the supplier to meet the specification will result in the rejection of the batch or the lower price. So, certain food do have certain microbes which are acceptable. They might not be pathogenic, but their presence might improve the flavor or be there for a particular reason. So, then we have to say that specification that the food can be accepted even if the microbe is present, but as long as it is in this threshold level, what we call specify the level and specify the microbe. So, the supplier accepts that whatever he produces, it will contain only less than that much amount what he has specified, or that amount, or not even less or less than or equal to. Anything beyond, he has to either face rejection saying that the batch contained extra amount of microbes which is not acceptable, or he might have to face a lower price. So, this is what we call as a microbiological specification. A microbiological guideline is used to monitor the microbiological acceptability of the product or the process. So, then you have what we call as a guideline. The difference from standard and specification here is that this is advisory rather than mandatory. Microbiological standard and microbiological specification are mandatory, has to be there necessarily if you are a food supplier, whereas a guideline is not. It is good if it is there, so it is advisable to have it, but it is not mandatory. The ICMSF specifications. This is a statement of the food to which criterion apply. So, for depending upon the food we are talking about, we are saying certain statement that because of these characteristics, the food can be acceptable. So, clearly the foods differ in their origin, composition, processing. They will present different microbial habitats and will therefore pose different spoilage and public health problems. So, as we saw earlier, depending upon the nature of the food product, plant versus animal, its deterioration nature, then its processing conditions, depending upon all these, the natural microbial population differ in each foodstuff. So, for each you will have natural population which is acceptable, then certain spoilage microorganisms which are specific to those type of food products. So, the type of these two will define what is the hazardous to the public domain when they consume the food concerned. A statement of the microorganisms or the toxins of concern. So, this would be different depending upon the different types of food. These may cover both spoilage and health aspects, but decisions on what to include must be realistic and based on a sound understanding of the microbial ecology of the food in question. So, what are the sort of microbes which are present, for example, in a cereal or a cereal flour powder, for cereal powder. For cereals, the water activity would be very less. We would be looking for more amount of fungi and molds rather than bacteria. So, there the spoilage organisms differ, whereas in the case of fruits and vegetables, we are looking about a different set of microbes which can spoil. The same in the case of marine or seafood, or in the case of animal products like meat, the type of spoilage organisms as well as the food poisoning organisms differ. So, when we say this statement, depending upon the type of food, this should be realistic. Details of the analytical methods to be used to detect and quantify the microbes and the toxins. So, by what methodology did you test for the presence of microbes? What are the microbes which you have identified? What was the identifying experiment, or what was the method? Or if it is a microbial toxin which you have quantified, by which method did you quantify the toxin? So, that has to be specified. Then preferred methods for standards or specifications would be those elaborated by international bodies, especially for food poisoning organisms as well as lethal toxins. You have international accepted methods. So, what you do and what you specify should preferably be those internationally accepted methods rather than going for some less sensitive or less reproducible methods. But in some cases, less sensitive or less reproducible methods may be used, especially for simplicity and speed in confirming the compliance with the guidelines. Sometimes it might take a longer time for the internationally accepted protocol to be done and for you to get the result. In such case, lesser sensitive and less reproducible methods can be used, but they are not generally accepted. The number and size of the samples to be taken from a batch of food or from a source of concern such as point in a processing line. So this should also be specified. What was your whole turnover? Out of that, how many batches did you or how many samples did you check for the particular spoilage organism, and what was your observation? So all those should be clearly stated. The microbiological limits appropriate to the product and the number of sample results which must conform with these limits for the product to be acceptable. So at the end of all these, the food can be accepted provided the microbiological standards is acceptable, what is internationally acceptable. Otherwise, food is liable to be rejected. For certain foodborne pathogens such as Staphylococcus aureus or Clostridium perfringens, their mere presence does not necessarily indicate a hazard, and specification of some numerical limits is necessary. Staphylococcus aureus, Clostridium perfringens and certain other microbes, their mere presence actually does not cause damage. Rather, it is the toxins of these which are lethal. So, if you detect the presence of the Staphylococcus aureus, it might not be a big concern provided the numbers or what we call the enumerated numbers remain less than what is acceptable. So, for there we have to specify that the numerical limits, that even though I detected Staphylococcus aureus, this was the detection number. Based on this, whether the toxins level might be high or low, or maybe if the organisms are live, they might be capable of producing the toxin which can be at the hazardous level. So, that numerical limit should be stated in these conditions. Microbial product quality or shelf life indicators are organisms and or their metabolic products whose presence in a given food at certain levels may be used to assess existing quality or better to predict product shelf life. So, we can use the microbes as an indicator of the quality of the type of food. So, either the number of the microbes or the presence of the microbes or the presence of microbial toxins would indicate, say that the food can be accepted or the food cannot be accepted. So, such things we call as indicator organisms. So, how do you evaluate that this particular organism can be used as an indicator organism? So, for that we have specific criteria. Number 1, the organism should be present and detectable in all foods whose quality or lack thereof is to be assessed. So, if I am saying that I am going to use microbe A as an indicator organism for spoilage or say pathogenic microbe present in a particular type of food, this indicator organism A should be present and should be detectable in all such type of foods. 2, their growth and numbers should have a direct negative correlation with the product quality. This is self-explainable. So, the number and the growth should be inversely proportional to the quality of the product. They should be easily detected and enumerated and be clearly distinguishable from other organisms. So, the indicator organisms should stand apart, should be easily detected, and they should be easily quantified, and they should not be submerged by the other microbes which might be present in the same food type we are talking about. They should be enumerable in a short period of time, ideally within a working day, which means that detection of the microbes have to be very fast. Their growth should not be affected adversely by other components of the food flora. So, even if the food contains multiple heterogeneous complex group of microbes, what you have chosen as an indicator organism, its growth should not be affected by the growth of the other microbial flora which are present in the same food concerned. The most reliable indicators of product quality tends to be product specific. So, what we take as an indicator organism should be specific to the particular product. So if I say that this indicator organism is present this much per milligram or per ml of this particular fruit, that itself should tell you that the food can be accepted or cannot be accepted for this particular product concern. Indicators of food safety. Microbial indicators are employed more often to assess food safety and sanitation more than quality. So we assess food safety using microbes rather than for quality. So first condition, it should be easily and rapidly detectable. It should be easily distinguishable from the other members of the food microbiota. The metabolic products may be used to assess and predict microbial quality in some products. For example, if you take a look about the metabolites which indicate the presence of microbes, applicable food products, the presence of cadaverine and putrescine in vacuum packaged beef, the presence of diacetyl in frozen juice concentrate, the presence of ethanol in apple juice fishery products, the presence of histamine in canned tuna, lactic acid in canned vegetables, the volatile fatty acids in butter and cream. All these, when they go beyond a particular limit, indicate that the quality of the food is compromised and it cannot be acceptable. The first organism which was identified to be an indicator organism was E. coli, and this E. coli was known as a fecal indicator. So when we talk about this we are going to talk about whether the food has been growing in a place where sewage has got mixed with water which is used for agriculture, or when you are using for processing, whatever is the water you are using for the processing method, has it been contaminated, or in the third aspect, whether the drinking water is potable or not. So there we look for fecal indicators. So how do we identify a bacterium to act as a fecal indicator? Ideally, the bacteria selected should demonstrate specificity occurring only in interstellar environments. So normally that organism which you have identified as a fecal indicator should have specific presence in the gut microbiome in the intestine. It should be there, specifically present in the human intestine. They should occur in very high numbers in fecus so as to be encoded in high dilutions. So when sewage is checked or when human feces is checked, those organisms should be present in very high amount so that even the highest dilutions will contain high numbers of this organism which will act as an indicator. They should possess a high resistance to the extraenteral environment and the pollution of which is to be assessed. So the moment they come out from the intestinal environment, they should be able to survive in the outside environment, resisting the hazardous substances there, which will indicate that yes, there is pollution here. And they should permit relatively easy and fully reliable detection even when present in very low numbers. So the detection of these should be very easy and it should be specific, so that even when at the highest dilution, even if it is present in very low numbers, they should be detected, saying that yes, there is contamination here in this particular foodstuff by sewage or fecal mixture. The first indicators we talk about are the coliforms. So while attempting to isolate the etiological agent for cholera in 1885, Escherichia isolated and studied the organism that we now call as E. coli. So since Escherichia discovered it, it is called as Escherichia coli. Originally it was named as Bacterium coli commune because it was present in the stools of each patient he examined. So he was looking out to find out what was the causative organism for cholera. So when he was assessing the stool samples of cholera patients, he identified this bacterium which he called as Bacterium coli commune. Schrödinger and other scientists was the first to suggest the use of this organism as an index of fecal pollution, as it could be isolated and identified more readily than individual waterborne pathogens. Later on, studies proved that E. coli is not the causative organism of cholera, but they identified that this is a normal normal flora in the gut microbiota. And Schrödinger then suggested that since this is present in the human gut and it is also passed out along with the human fecus in high number, this can be used as an indicator organism to find out whether there is fecal pollution in food components, and it can be easily isolated and identified rather than the other waterborne pathogens. This normally the presence of E. coli is used to test for the potability of drinking water. The gram-negative rods that ferment lactose within 48 hours and produce dark colonies with a metallic sheen on endotype agar. So this is actually the test what we do for testing the potability of drinking water. E. coli is generally a gram-negative rod. It is a lactose fermenter within 48 hours. So when you grow it in a lactose containing medium, it will ferment it, and then on an EMB agar on an NB you see dark colonies with a metallic green sheen. There are it is represented by 4 genera of the family. The coliforms are represented by 4 genera of the family: Enterobacteriaceae, Citrobacter, Enterobacter, Escherichia and Klebsiella. So these are the 4 organisms, genera, which we talk about, which belong to coliforms, which can be considered to be a fecal contamination indicator. The IMVIC formula is the classic method used, where I is the test for indole production, M is the test for methyl red reaction, V is the Voges-Proskauer reaction, where we look for the production of acetoin, and C for the utilization of citrate to the carbon source. So depending upon whether the organisms answer these tests positively and negatively, we can say that this is the particular organism which is in the contamination zone, which contaminates the foodstuff or the drinking water. So for example, using this IMVIC test, 2 organisms have the following formulas. For example, E. coli, it is IMVIC indole positive, methyl red positive, Voges-Proskauer test negative, citrate utilization test negative. Whereas Enterococcus aerogenes is indole negative, methyl red negative, Voges-Proskauer positive, citrate positive. So depending upon the result you observe among the Enterobacteriaceae, you can identify which is the particular organism we are talking about. So what we do for a testing is, we first serially dilute the food homogenate and then inoculate in tubes, LS tube, lactose tube, broth tubes, incubate at for 35 degrees for 48 hours. If there is, when lactose fermentation happens, gas gets produced. So depending upon whether the gas gets produced or not, you identify and separate out the gas positive tubes. Then in the gas positive tubes, on one hand, we will do an MPN presumptive test for total number of coliforms. From all the gas tubes, we will inoculate in the brilliant green blue bile agar or the broth and incubate at 35 degrees centigrade for 48 hours. Then again look for gas production, and in the BGB tubes which show gas production, calculate the MPN from the accepted mass. Then we have the test MPN test confirmed for the total number of coliforms here. On the other hand, to test for the fecal coliforms, we will inoculate this in the easy medium and incubate at 45 degrees. And those tubes which show gas and the tubes which will, for this you will look for the total MPN for the fecal coliforms. On the other hand, if we inoculate in the lactose with MUG, methyl umbelliferyl glucuronide broth, and incubate at 35 degrees centigrade for 24 to 48 hours and examine it under UV light, those tubes which show fluorescence. So we will classify here as a fluorescent positive tube and a fluorescent negative tube. This fluorescent positive tubes will contain E. coli. So based on this, we can identify the total coliforms, the fecal coliforms and E. coli. So this is a normal test which is done in any food testing lab. The coliforms grow well on nutrient agar and they produce visible colonies within 12 to 16 hours at 37 degrees centigrade, and these can grow in a large number of food under proper conditions, and they are capable of growth in the presence of bile salts. Normally the presence of bile salts will inhibit gram positives, but the gram negative coliforms can grow even in the presence of the bile salts. So what is the standard and what is the criteria? Under proper conditions of harvesting, handling, storage and transport of foods by use of a hazard analysis critical control point, otherwise called as the HACCP system, the lowest possible and feasible number of coliforms. So how do we say that under this HACCP system, what should be the lowest possible amount of the coliforms which can be present in a particular type of food? Then 2, at quantitative level, how do the levels of coliforms indicate that the product has become unsafe. So that is what we will now look for, and look for the product quality. So under the HACCP system, the presence of the coliforms should be not more than 10 per ml for grade A pasteurized milk and milk products, including cultured products, not more than 10 per ml for certified raw milk, and certainly not over 1 for pasteurized milk, not over 10 per ml for pre-cooked and partially cooked frozen foods, not over 100 per ml for crab meat, and not over 100 per ml for custom fill item. So these are the accepted standards. If the number go beyond this, the food cannot be accepted under HACCP. So low numbers of coliforms are permitted in sensitive foods at numbers ranging from 1 to not over 100 per gram or 100 ml. This criteria reflect both the feasibility and safety parameters. So in this lecture, we saw how coliforms can be used as a microbiological indicator of the safety as well as the quality of the particular food concerned. And in a food testing lab, we also saw how the test for the presence of coliforms is done and what is the acceptable level. In the next lecture, we will see what are the other indicators of microbial indicators which can be used to test for the quality of the food, and what is the procedure for that. Thank you.